Yeah, we saw.
Yeah.
Some of the problem last week, the AV person.
Oh, I only went for a tiny bit.
That was
So hopefully we're not going to see that anymore. And these guys.
They're in Portland, so they drove up.
Did one of them do it? We're going to be in good hands here.
Let's go ahead and get started. The good news for those of you listening from the outside, you'd probably be skeptical about what I'm going to say, but we actually think we know how to broadcast these sessions correctly, both the audio and the video. So anybody who's listening, Mike Weiss in particular, if you don't hear the audio, text me this time so I know, but I'm pretty sure from in-house investigation that this is working. And we know how to do it going forward for the rest of the year. This morning's speaker is Steve Ziegler from Benaroya Research. I thank him very much for coming and giving us this talk, which I'm sure will be very instructive and educational for all of us on epithelial cytokines and their role in allergic inflammation. Steve, thank you.
No problem. Thanks for the invitation. I'm not used to being functional at this hour. I will try. So what I thought I would do today is go over a series of vignettes of work from our lab that has, over the years, looking at the role of cytokines from the airway epithelium and how they control responses to various allergens, pathogens, what have you. And the sort of overarching theme that I hope I get across is that it's very varied. The response to cytokines depend on what the actual trigger is, and that the downstream players that are involved in the response are also very different, depending on whether you're talking about house dust mite, cockroach, allergen, a respiratory virus, and it's all the same sort of group of cytokines that are involved, but it's hard to predict who's going to be involved just by saying, you know, we're going to look at house dust mite, for example. So I'm very boring. I have no disclosures. So this is basically what we're looking at. This is an airway epithelial barrier. It's getting bombarded by a variety of different things, which happens all the time. Well, it's not really helmets in the mouth usually, allergens, what have you, and what they all tend to have in common is that they elicit a coordinated response from the epithelium that results in the release of a variety of cytokines. The three that my lab has been most involved in are these three here, which are IL-25, IL-33, and TSLP, which stands for thymic stromal lymphopoietin, which is the worst named cytokine in our lexicon. It actually has nothing to do with the thymus. It just happened to be isolated, in the late 80s, early 90s by Andy Farr when he was still here, from a thymic stromal cell line. But it's actually expressed, as I'll get into later, by barrier epithelial cells, and is important for driving a type 2 response. So these, and before I get going, I used to be of the belief with the epithelium that it's just a stupid barrier, that it's there to keep things out, but it really doesn't do anything. You know, all the really good stuff is done by, you know, the immune cells that are below it and in the interstitium and in the circulation. I've now, after studying these cells for 15, 20 years, have come to the conclusion that they are actually an innate immune cell. They are the brains of most of the responses that occur at their areas, be you in the airways, in the esophagus, in the gut. And they coordinate the kind of response that will happen downstream by what the pathogen, either real or perceived, is. And so they sort of control everything that's going to happen later by what they release, be it a cytokine, be it a lipid mediator, what have you. And so they're very complicated cells. fascinating to look at them. They're totally different whether you're looking at them, at least in the lab, as a submerged culture or as a polarized culture, and they tend to live polarized. So don't ever believe any data that you see from people using submerged cultures of airway epithelial cells or colonic epithelial cells, because if you look at the same response in the same cells that are grown in that way or grown as a polarized culture in the air-liquid interface, if you're looking at airway or colon, it's totally different, completely different. You'd think you're looking at two totally different cell types. And so I've rejected many a paper for them using submerged cultures rather than polarized cultures. It's a lot more difficult to do polarized cultures, but it's reality. So I do digress. Anyway, so what happens is these cytokines get released, and they, whoops, sorry about that. And they activate a variety of different cell types that are living just below the surface there. These innate lymphoid cell type 2s, which are basically a T cell without a T cell receptor. They do all the things T cells do, except they do more of it. They make pound for pound, they're probably the biggest cytokine producers in your body. There aren't very many of them, but they're really potent, and they are the sort of priming cell for most of these responses. They make cytokines, IL-13, IL-5, IL-9, that sort of drive downstream responses from a variety of cell types: eosinophils, mast cells, basophils. You get these cytokines can also activate dendritic cells, which can then go to drive Th2 or Th, depending on what the response is. T cell differentiation, either in situ in the lung, or in the migrate to the draining lymph node and drive differentiation in the lymph nodes, and those cells come back. Either they go to germinal centers to drive IgE production, or they come back to the lung and drive the response that's there. So this is sort of the environment that we'll be living in today, and we'll go forward from there with specific examples. I'm going to give a few examples of how we do this. We look at things like house dust mite and cockroach allergen, which are very common allergens here and around the world. I'll give a little bit of work that we've been doing collaboratively with Jason Debley's lab at Children's on the response of human airway epithelium to respiratory viruses, just to give you an example of how these cytokines are induced by these viruses. And then, not on here, but I'll close with an interesting story on food allergy and how skin sensitization can drive responses either in the gut or in the airway, depending on where the subsequent challenge comes from, and then who's responsible for that, for driving those responses. And so this is our model of house dust mite airway allergic inflammation. And we give it's a relatively mild model compared to what you'll read in some papers. So we wanted to do it so that we could actually see what was going on without a sort of sledgehammer effect of having a lot of dust mite around. So we do a single 1 microgram sensitization challenge, which is basically, and this is all done intranasally. We wait about a week and come back with 3 consecutive challenges of 10 micrograms and then do the response. And you can see the response is still pretty potent. In the PBS control, you can see that there's basically the only cell type in the BAL, and this is all cells from the bronchial alveolar lavage, are alveolar macrophages. They're about 95% of the cells that you get out of a BAL of a naive mouse. But after the dust mite sensitization challenge, we see that most of the cells are now eosinophils, these cells in the upper left quadrant. And they're about, depending on the day, the time of the year, whatever, they're between 70 and 90% of the cells that we get out of the BAL. We don't lose alveolar macrophages, so that it appears that there are fewer of them, but just as a fraction of the total cells you get out of the BAL, they go from 90% to 10 or 15%. And some of these mice, you can see here when we do total cells, this was a sort of a weak response. There's only about 150,000 cells. our record for the number of cells we've got on a BAL was 13 million. The mouse basically exploded. But, and we can sometimes get somewhere between 3 to 10 million cells out of a BAL, and 80 to 90% of them will be eosinophils.
So the marker for that is cyclic F?
And yeah, they're cyclic F high and CD11C negative.
So last week's speaker talked about cyclic 8 in humans. Is that the same thing? The mouse marker is cyclic F?
It's sort of the same thing. Mice don't have cyclic 8 and humans don't have cyclic F. But they sort of fulfill the same functionality.
They're markers for eosinophils.
For eosinophils. Now, mind you, we have no idea what these eosinophils are doing, but they're very, very good because you can do this in an animal that lacks eosinophils genetically, and other than that whole pile of cells in the upper left quadrant, everything else is the same. And so we sort of use them as our marker for a type 2 response in the airways, but it's still unclear what they're actually doing. But you can see there, most of the cells that come in are EOs. And we tried to figure out what's going on with cytokines. We looked at TSLP deficient mice, we looked at IL-25 deficient mice, no change whatsoever. But when we looked at IL-33, what we found is first of all, dust mite induces the expression of IL-33, both at the RNA and more importantly at the protein level. And IL-33 is an interesting cytokine in that you usually, when it's involved in a response, you usually don't see much of a transcriptional change. And 33 is basically made constitutively by these cells, and it lives in the nucleus. And it gets released by cells as they die. So sort of as a response, they're being killed because they're infected with a virus or they're responding to an allergen, and then 33 gets released in some mysterious way. There are also other pathways, other cell types can make IL-33 monocytes that come into a site of inflammation, can make it. It's not clear what secretory pathway they use, but they don't die. So 33 is a idiosyncratic cytokine. So it's much more important to look at protein expression rather than mRNA expression. But here you can see there is about a 2-fold increase in transcription and about a 10-fold increase in the amount of protein that's made or released into the BAL. And when we look at the response, and who's making this IL-33, this is looking at a wild-type mouse in dust mite. In the middle is IL-33 deficient mice, so they make none at all because they don't have the functional gene. And the last column, which is the important one for the rest of this part of the talk, is a mouse where we have a conditional knockout allele of IL-33, so it's floxed. It was given to us by a colleague at Northwestern, Paul Bryce. And the CRE driver is surfactant protein C, so it's in the epithelium. And this is a construct made by Jeff Whitsitt about 25 years ago at Cincinnati Children's. And Jeff designed it so that it's not only expressed in cells that normally make surfactant protein C, so alveolar type 2 cells, but they also, it's pan airway epithelium. So every epithelial cell in the airway will be deleted of whatever gene is floxed in that mouse. So here we have about a 90% decrease in the amount of IL-33 that's made if you knock out. Yes.
What does floxed mean?
I mean, so it's a system now is co-opted from bacteria, where there's a DNA sequence called a LOX-P site, and the bacteria make a enzyme, a recombinase called CRE, and basically CRE recognizes the LOX-P site and cuts. And so if you flank a gene or an exon, what have you, with LOX-P sites and express CRE, it will delete what's in between those two LOX-P sites. And so it's a way that has been developed over the last decade or so to get conditional knockouts. So you drive this CRE, you have every cell in the mouse has a floxed allele of IL-33, and it doesn't affect the expression of the gene, the function of the protein at all. The only cells that are going to lose expression of IL-33 because it gets deleted here are airway epithelial cells. If you use, for example, a CRE driver downstream of CD11C, you get deletion in myeloid cells for the most part and some NK cells. You know, it's a way to knock out a gene specifically in a given cell type or cell lineage without affecting it broadly. And to add another twist to this, there is a way to make the CRE conditional. So you make it a fusion with, oh God, I'm failing on this. With another protein basically, and then you add tamoxifen, and that releases that, and then you get CRE activated, and then you can get. So that's a way to do it. Some of these knockouts have developmental issues, which you're not really interested in, you know, necessarily. So you want to wait until the mouse is an adult before you knock out the gene. So you use these conditional CREs to do those experiments. So it's a way that's been developed to basically knock out genes.
Well, you just alluded to what I was going to ask you. Is anything wrong with these mice? Let's say the 33 knockout mice, are they normal?
The straight up knockout,
Yeah.
they're not normal.
They're not.
So 33 is also a really interesting, it's a huge target now for biotech companies and pharmaceutical companies for reasons like that I'm going to show you in a moment. But it also is very positive effects in say, heart attacks, things like that. It helps repair after infarcts. It's involved in cognitive function. So it's important when your brain is developing, it's involved in trimming neurons. It activates microglia. And so mice, for the most part, are not very bright, but the IL-33 knockout mice are even stupider than a wild-type mouse. And so I have a little bit of an issue with, you know, anti-33 drugs for long-term chronic use. Although hopefully in an adult, their neurons are trimmed. But I wouldn't want to have a heart attack while I was on anti-IL-33. But otherwise, they're fine. They have some white brown fat problems because IL-33 is important for ILC2s that live in adipose tissue. But these mice are all under SPF, so they're not going to be challenged much. We are setting up, just as an aside, we'll be testing all these things because we're setting up a sort of mini colony within our mouse colony with wild mice that bring in a real bona fide microbiome, and we'll see what happens in those instances if those mice still are okay or if they crump.
Steve, you were talking about the importance of polarization
Right.
of epithelium. Is there spatial differences in the IL-33 expression? Could that go with? What is the main difference of polarization?
We actually haven't looked. Other people have used reporters for IL-33, and it looks like it's pretty much pan-epithelial. If you look in the airways, both in the bronchus and in the alveoli, so it looks like it's pretty much everywhere. But if you look at most places where IL-33 is expressed, it's in every cell that's there. It's, you know, you look in the colon, and every colonic epithelial cell is positive for IL-33. It just doesn't get released until it needs to be. And so if we use this SPC CRE IL-33 flox mice, you can see that basically we lose most of the response to house dust mite, as read out by these eosinophils. And if you look at the histology of these mice, they lose most of the sort of histological features of the response. There's not goblet cell metaplasia and mucus overproduction. You lose most of the infiltrates. And so IL-33 is clearly driving this response, but we wanted to try to ask who's responding to IL-33 downstream of the epithelium. And so, and this is just looking at it, that you see about a 90% decrease in the response because you have only 10% of the amount of IL-33 around. This is looking at total cells and eosinophils on the other side. And so we want to see who's driving in this. And so we started looking at cells who are expressing a protein called ST2, which is the main IL-33 binding subunit of the IL-33 receptor. And these are cells that are CD11B positive. Is that B or C? I can't remember. I think it's B. So they're monocytic cells. The cells that are positive here that are staining with an antibody against ST2 are also MHC class 2 positive and CD11C positive, so they're basically dendritic cells, and antigen presenting cells, and these are out of the lungs themselves, so in the lung parenchyma. And if we take a different mouse that we made in my lab, which is a floxed allele of ST2, so it's just the opposite. Now we can conditionally and very specifically remove the receptor for IL-33 and therefore either change or block the ability of particular cells to now respond to IL-33, not to make it, but to respond to it. And here you can see that you get a nice response. It's not great. This is work that was done by a rotation student in the lab a couple of years ago. But basically, if you make a CD11C CRE ST2 flox mouse, which basically you're eliminating dendritic cells as a responding cell type for IL-33, you lose the response. So the take-home lesson is that what we think is going on is that, dust mite comes in. This is your airway epithelium. Dust mite comes in, activates IL-30, turns on IL-33, releases it, and you drive dendritic cells to drive Th2 polarization, we think, and that drives Th2 differentiation, eosinophilia, and other responses that we're still trying to understand right now. We're now, sort of walking through the cells that we know are in the lung and knocking out ST2 and T cells and B cells in monocytes, things like that, to understand what the sort of hierarchy of the response is. But this is where we're at right now. So dust mite is IL-33. We've now started looking at other allergens. So we're an equal opportunity lab as far as allergens go. So now we're doing some work with cockroach allergen, and it's you just buy it from a company and you stick it up a mouse's nose and see what happens. And so this is the model that we're using. It's a slightly different model. This cockroach needs a little bit more sensitization to get a good response later. So we have 3 consecutive challenges, and I believe this is again with a microgram each time of cockroach allergen. We wait 2 weeks this time and then give 2 challenges of I think it's like 25 micrograms of cockroach, and then look at the response. And we get, sure.
So this is one of the places, this is just a clinical question, one of the few places in the world where we actually don't see human beings sensitized to cockroach. And so
They don't like the cold and the wet.
Huh?
They don't like cold and wet.
I don't know. I never asked them. But we don't have many of them here.
Okay.
So I'm wondering, these are of course experimental mice.
Right.
And if you got rats out of the sewer here, would they be cockroach sensitized?
Probably.
Well,
I don't know.
Humans aren't.
We don't
so
know.
I don't know.
Yeah.
Because most people in the rest of the United States are
Especially in urban areas, yeah,
sensitized,
yeah.
But we don't see them here. Right.
What is the main allergen? Is it
Well, I mean, dust mite is
yeah.
very dramatic, very important
Yeah.
here, but, inner cities and the
Right,
rest of the
it's
United
all
States,
cockroach, yeah.
cockroach is
Right.
truly a major
Yeah.
allergen in driving allergic disease.
Right. So basically, you get a really nice response, and again, you can see it's largely eosinophils that are coming into the lung in these animals. Although you do get a nice CD4 T cell response, nice neutrophil response, which will be interesting later on. We still don't understand that, but that suggests, because neutrophils are usually recruited by cytokines like IL-17, so a type 17 response rather than a type 2 response. So this would argue that what cockroach is driving, at least in this model system, is a mixed type 2, type 17 response, and we still don't understand, and I'll get to that at the end of this little part of the talk, who's making we assume it's IL-17, we haven't proven it, but who's making the cytokines that are attracting the neutrophils and what the neutrophils are doing. But it's another example of a different allergen delivered the same way, a different kind of response.
You think that's because of what is in the allergen? I mean, it's very high LPS.
Right, no, because we worry about that a lot, because Matt brings up a good point, is we get these from this company called Greer. They basically just, in this case, take a cockroach, in the other case, they take, dust mites and grind them up and just sell them as a powder. And there is endotoxin in there. And so what we usually do is we measure the amount we buy a big lot, so that with all the experiments we're doing, we use the same lot of whatever the allergen is. And we measure the amount of endotoxin that's in that lot. And then we do a set of animals where we give them that amount of endotoxin without the allergen to ask, what does the endotoxin do? And in most cases, it's low enough where it does nothing. Or we'll do the experiment in a mouse that lacks TLR4, which is the receptor, if you will, for endotoxin, and look at the response there. So that's a good question. We don't think the endotoxin is doing anything. And we've done it. We know in this model that at least at a gross level, I'm sure it's doing something, but at a gross level, it's not driving this response.
Can you tell us what the receptor on the epithelial cells for these antigens is?
So it's interesting. So for dust mites, it's a complicated thing. It's part TLR4, it's part 2, so protease activated receptor 2, which is a very interesting receptor that's on the apical side of most epithelial cells. And it's a receptor that both encodes within its protein structure a receptor and its ligand. But in its normal state, the ligand is attached and doesn't affect it doesn't trigger the receptor. But if it gets cleaved, it then acts as a ligand for that receptor and activates. And most of these allergens have protease activity, and they activate. Certainly cockroach does, dust mite does, Alternaria is another allergen that we've used that activates part 2. And then part 2 signals drives NF-kappa B activation within the epithelium, and cytokines that I'm talking about here are all exquisitely sensitive to NF-kappa B. So TSLP, for example, has 2 NF-kappa B sites, and it's in an enhancer that's a couple kb away from the start of transcription, and it's as soon as it sees NF, boom, it's off. And so that's largely what's driving this, but there are also toll-like receptors that are involved in the response as well. And so this is just looking, it's largely a type 2 response. So we get, with PAS staining, we can observe goblet cell metaplasia, mucus overproduction. We get expression of IL-4, IL-5, and IL-13, the sort of, type 2 cytokine world. But we also, as I mentioned earlier, we also get IL-17. We don't get a type 1 response, so there's no gamma interferon response in this to cockroach. But we get a pretty healthy IL-17 response. And then again, I will preface this by saying we don't know who's making the 17. I suspect now that we've done a few other experiments in other model systems, one type of cell that I'm not going to talk about other than right now, that gets recruited in these types of situations are gamma delta T cells, sort of the other T cell. And they make a lot of IL-17 in these model systems. So I suspect, with no data to back it up, that it's gamma delta T cells that are making this IL-17. What its role is, we have no idea. We're still looking at that now. And so we wanted to ask, what cytokines are driving this, with the hope it would be one of our big 3. It's not TSLP and it's not IL-25. These are basically looking at the response to cockroach in either a wild type mouse or a mouse on the left that lacks the TSLP receptor, or on the right that lacks the receptor for IL-25. And you can see the response either in total cells that are infiltrated into the BAL or eosinophilia is unchanged by not responding to these cytokines. So it's not any of those. When we looked at IL-33, we did see a diminished response for eosinophilia, so it may be involved in that aspect of the response. But when we looked at other aspects of the response to cockroach, so the infiltrating T cells, dendritic cells, and neutrophils, IL-33 was not involved. And so for the total response, it's not one of our favorite 3 cytokines, which was kind of a bummer. because then we didn't know what the hell to do. And so we went to the literature. This was a really talented graduate student who graduated a couple years ago. She's now a postdoc at the Hutch. Dug into the literature and asked, what else do people see with cockroach? And it was a lot of it was human work. And it turns out that we started looking at other molecules that might be involved in this response to ask, can we understand who's involved in this by looking at what signaling pathways are important for response. So MyD88 is an adapter molecule that's attached to toll-like receptors and receptors for IL-1 family members, so IL-1, IL-18, IL-33, IL-36, etc. And when we knock out MyD88, we lose most of all of the responses. So you can see both the eosinophil response on the left and the other cells that come in on the right panel are knocked out pretty much by MyD88 deficiencies. Likewise, the cytokine responses across the board are lost. So clearly, whatever's driving this response is using MyD88 in some way to drive the response. So that gave us a clue. It could still be IL-33 for part of it because MyD88 is involved in ST2 signaling. And then we wanted to see whether the epithelium was involved. And so we used the same SPC CRE, and in this case, we used a floxed allele of MyD88, so we could only knock it out in the airway epithelium. And we also lost the response. So now we know that it's signaling by the epithelial cells that is important for driving MyD, and that eliminated IL-33 because you don't need MyD88 to transcribe the IL-33 gene. It's involved in signaling, and we don't think that ST2 is signaling on the epithelium. The epithelium doesn't make ST2. And so we thought, well, what the hell's going on now? It's driving, so somehow MyD88 is driving the expression of whatever factors, whatever they were, that are driving the eventual response to the cockroach. And so this is where Alyssa went into the literature and found that, in fact, GM-CSF, which is another cytokine that tends to be thought of as a T-cell-derived cytokine, it's a cytokine that's important for driving responses that T cells do. It's important in MS. It's probably the pathogenic factor in multiple sclerosis, and more importantly in EAE in mice. But it's involved in the airways. It's been shown to be important in asthmatics, as you can see here. And so we asked, well, is GM-CSF somehow involved in this? And so we did the same experiment, looking, but instead of extending it for the whole 2 or 3 week time, we asked, does cockroach even induce GM-CSF expression? So we basically took a mouse, gave it cockroach intranasally, waited 2 hours, sacked the mouse, and then did BAL and the lung tissue for RNA, and asked, do we see GM-CSF induced? And the answer, of course, is yes, or I wouldn't be showing you this. So we figured that GM-CSF at least is induced by cockroach, and we know from the SPC experiment we suspected it was the airway epithelium making it, which was kind of surprising to us. So we did what I'll be coming to later, these air-liquid interface or polarized cultures of epithelium. You can do it from humans, you can do it from mice. Where basically you grow them on a small transwell. You let them basically go to about 80% confluence, and then you change the media and put them in this kind of a configuration, and the cells polarize to have an apical and basolateral surface. And these are nice because you can apply allergens, viruses, what have you, to the apical surface and then read out the response from the basolateral side. You can put other cells in the culture and ask, who gets turned on by, in this case, we could put T cells, APCs, what have you, and we do this a lot now with human, with Jason and Matt is involved in this. Looking at responses from cells that are in contact with epithelial cells that are now being challenged by various allergens, but in this case, we're just doing this to see who makes GM-CSF, and you can see that in these cultures we get nice GM-CSF expression if we challenge them with cockroach. If we do this from epithelial cells from a MIDI88 knockout mouse, we don't. So clearly MIDI88 is involved in allowing cockroach to turn on GM-CSF transcription. And if we do it with the SPC CRE, we see the same thing. So clearly this is cockroach allergen triggering some receptor, could be PAR2, could be TLR4, and we now know that it is TLR4, turning on MIDI88, which then drives a response which we're not sure what the transcription factors are that turn on GM-CSF transcription. And so, is this important? You know, it turns on GM-CSF. You can't. We would do this the easy way if we could with a GM-CSF knockout. But the problem is GM-CSF in the lung is really important for alveolar macrophages. They need it to live. And if you knock out GM-CSF, you get alveolar proteinosis, and the mice die. Same thing in humans. Likewise with a GM-CSF receptor knockout. So we couldn't use the knockout because there's obviously problems with that. So we just did the next best thing and used a blocking antibody. Basically treated the mice with anti-GM-CSF and then did the whole response as you can see. And treating with it knocked out everything as you can see there. So we don't get eosinophils, we don't get CD4s, we don't get DCs, we don't get neutrophils. So the whole response is gone if you treat during sensitization with anti-GM-CSF. However, if you treat during the challenge phase, there's no effect whatsoever. And so what GM is important for here, and differently from what I showed you in the dust mite response, is GM-CSF sets things up. It basically primes the response in the lung to respond to a second challenge with cockroach. But it's not involved at all in the response to that second challenge because it's already done its work. It's already primed the immune system, if you will, to recognize cockroach the next time it sees it. And so what's it doing? It's basically, like I said, it's priming the response. So we did this experiment where we did the anti-GM or not, just during sensitization, and then sacked the mice afterwards and asked what was going on. And basically what we see is a loss of IL-4 and IL-13, suggesting that what it is doing is it's preventing T-cell priming. Basically, if you block GM, you don't get appropriate Th2 priming, presumably in the draining lymph node, which is what we found. So we did this. We took cockroach that Alyssa had conjugated with a fluorophore, Alexis, in this case it's Alexis 647. And what that allows us to do is follow cells that take it up, because they'll take up the cockroach, but they also take up the fluorophore. And so you can then do flow cytometry and ask, okay, if we look in the lung and if we look in the draining lymph node, do we see cells that have taken up cockroach in the lung and have now migrated to the draining lymph node to start the response? And what we see is that if we look at wild-type mice, and this is looking at Alexis 647 responses and CD80, and CD80 is a co-stimulatory molecule that gets induced on antigen-presenting cells when they take up antigen and they're ready to prime a T-cell response. And you can see two things here. One is there's about a roughly 50% reduction in the number of cockroach-positive cells that make it to the draining lymph node if you block GM-CSF during sensitization. And the expression of CD80 is reduced quite considerably on those cells that are from the mice that got GM-CSF blockade versus the wild-type mouse. So you get fewer cells that are less able to drive a response in the draining lymph node if you block GM-CSF in this case. And we see, and it's all quantified there. We get fewer cells by frequency and by number, and the MFI, or mean fluorescence intensity, which is a measure of the amount of CD80 that's on, in this case, on the cell surface, is down, 3.5-fold. And most of these especially these co-stimulatory molecules, they have thresholds that, you know, you need to have to get a proper response. And this is below the threshold. And so GM is basically setting up sensitization, but is not involved in the challenge once sensitization is done. And so this is how we think it's going. I didn't show you the data, but it is TLR4 that's driving the response on the apical side of the epithelium. It's triggering through MIghty88 the expression of GM-CSF, which is then activating resident dendritic cells in the lung. They take up the allergen, in this case cockroach, migrate to the draining lymph node and drive T cell proliferation and Th2 differentiation. We don't know what IL-33 is. We're checking right now, but I suspect what IL-33's involvement in this response is in the challenge. It may be involved in re-triggering the response to cockroach once GM has already done its work. And what we did do, and I probably should have put the data in because I was worried about time. What we did in the case of the MIghty88 knockouts is we gave the mice GM-CSF when we gave them cockroach allergen. We just, you know, put it in the mix and put it in, and we could recover the response quite nicely. All of the response except the IL-17 response. Yes.
In this model, you keep referring to cockroach. Is it a generic model for sensitization, or is this just unique to cockroach?
The GM part of it, it's just cockroach. So it's the essential model that I'm showing here is the same one we use for dust mite, and the same one we use for Alternaria. And whatever allergen we're using, we do intranasal sensitization and intranasal challenge. The response is specific for a given allergen, and they're all different. Each one of those they're different. They're different in the cytokines that they elicit. They're different in the cells that are important to drive the response. And I guess that's one of the big take-home lessons is that there's no single model that you can put together for allergen does X.
Is that because they start with a different TLR or a different?
Right. The components of each one are different. You know, some of them are proteases, some of them are other molecules that drive TLR activation. There are other allergens, and I didn't even get into this, that also trigger things like arachidonic acid metabolites that are important for some responses, especially for ILC2s. And so it's complicated. I didn't want to get too much into the weeds for this. I was trying to focus on both the cytokines that are involved and the diversity of the responses. Yes.
So what about so there are like IRF4 patients and MIghty88 deficient patients. Do you see this in them the same way that you see it in?
Probably, but they have so many other problems that I don't think they're going to be worried about being allergic.
So I mean, the theoretical thing with them is
Right.
once then they're older. So I mean, like, you know, they can get very sick
Right.
when they're little and septic,
And then
but
they
if they make it through that, then usually they do well. We had 3 IRF4 patients where I did fellowship that were not related, but then they started to have a lot of like chronic rhinosinusitis
Right.
polyps.
Yeah.
But I'm pretty sure one of them was alert. Like they also had like underlying allergies too that had developed, even though theoretically
Right.
most of their pathways were not working in that sense.
Yeah, interesting. Matt.
I think there is literature that population generally does not develop allergies. I think I've seen that for the CIS. But on that as well, so do we know what part of the allergen signals through TLR4? Because TLR4 will signal through MIghty88
Right.
and then a separate pathway as well. Why do you think MIghty88 is so critical in this model, given that TLR4 has multiple signals?
I don't know. It's just the observation. We didn't do TRIF knockouts to see if that pathway is also involved, because the MIghty88 TRIF double knockouts are really difficult to deal with. And we got a response with MIghty88, so we didn't bother. Any other questions? Great. And so now TSLP, the best cytokine in the world. I've been
Question
studying.
before you get into
Sure.
TSLP, you know, down the just as the theoretical precision medicine, inner city kids, I mean, cockroach is a
Is
big
important.
problem. If you were to design an intervention there that would help them, but knowing the cascade, what would be the?
It would be hard to do anti-GM because you're going to have issues with alveolar macrophages. If you could show that IL-33 was involved in the challenge response, because most of those kids are already sensitized, so the GM won't matter. So potentially, anti-IL-33, which has its own issues. You could also think about the effector arm of the response and treat with dupilumab. You could take out IL-4 and IL-13, and that's probably the way I would go because it works. And there are trials now in asthma, and they look pretty good. They're not as good as the TSLP trials, but they would work in that case probably. So I would probably do that. And it's great for Regeneron because then they got patient for life because as soon as they go off drug, it comes back.
Is that your theory without, obviously, the data that all the sensitization pathways are different, but then they have the similar effector pathways
Yeah.
after that?
Yeah, it's very stereotypical afterwards. It's all type 2. But you get there differently depending on what the allergen is. And for you guys that are in the clinic, once you see patients, they're already sensitized. So you're just dealing with, the effector arm of the response. So TSLP, it's made primarily by epithelial cells at barrier surfaces, but it's made by a bunch of cells at sites of inflammation. It generally activates myeloid-derived dendritic cells, but a lot of other cells can respond to it depending on the type of response. And the observation that got this field going about 20 years ago by Young-Jun Liu when he was still at DNAX was that patients that have, and I'll show you this data in a moment, with atopic dermatitis, if you look at lesional skin in those patients, they're blazing hot for TSLP. Adjacent, uninvolved skin, it's barely detectable. In a healthy individual, it's barely detectable. But in lesional skin, it's smoking hot. And if you look now in the lungs of asthmatics and the noses of patients with rhinitis, there's lots of TSLP. So we think it's basically the major underlying factor in all atopic disease. And you probably know this better than I do, but there's really amazing data with an anti-TSLP drug that's in now starting phase 3, but the phase 2b trial in asthma, all comers, Th2 high, Th2 low, neutrophilic, and looking at exacerbations over a year, it worked in every patient, down 75% over a year, regardless of what kind of asthma you had. And so it looks really promising. In our models, not so much. So the receptor is a heterodimer of the ligand-binding TSLP receptor subunit and the IL-7 receptor alpha chain. And as far as we know, it only signals downstream to STAT5. And that's all of the response. This is that trial that I just mentioned, co-run by MedImmune and Amgen, 3 different doses over a year. And the data look like that. Basically, at all doses in all patients, we saw dramatic they did. I wasn't involved in this. I'm sort of on the mechanistic side of this. And I collaborate with both Amgen and MedImmune to get samples. But basically, it worked across the board. And so they just set up and they're recruiting now, and I think they're almost finished. Most of this work is done by Jonathan Corin down at UCLA. And it's pretty remarkable. So anyway, so it does do something.
What does it do for us other than revive atopic disease? What's its benefit?
It's unclear. There are,
Knocking it out?
so you knock it out in a mouse and they're in an SPF mouse and they're fine, as far as we can tell. They have a few fewer T cells and things, but they're generally okay. There are humans that are TSLP knockouts. We know that from looking at the Icelandic database, where most of Iceland's have their genome sequenced, and you can search on genes to see if there are knockouts and what have you. And there are plenty of people that are either receptor or cytokine knockouts, and they're perfectly fine as far as we know. So it's not clear what it does normally. We think what it does. And again, this is hypothesis with no data to support it, but it's made by, at barrier sites that are, susceptible to getting bombarded. So in the airways and in the gut. And what I think it does normally, it's low-level expression, and it creates sort of a type 2 homeostatic response around barriers, which keeps them quiet. Because around a barrier, you don't want to have a type 1 environment, because type 1 responses tend to be explosive. Type 1 responses basically kill cells, and you don't want that at a barrier. You want to have a sort of a type 2 sort of friendly response there, and I think normally that's what TSLP does. We also know that it's important, now we have new data, that it's important for some germinal center responses. So it may be involved in maintaining sort of antibody responses to commensal bacteria in the gut. We're looking at that right now. But it's not really clear what it does that's good. We know a lot about what it does that's bad. It's just one of those situations where it's hard to do with the experiment to find out what's going on. We're also going to, what we call pet store, the knockout mice, basically put them with wild mice to get a wild microbiome and see if that affects their health. So this is one of those ALI cultures. This is actually a picture that Jason Debley took. This is from a human bronchial brushing. Was the source of the cells. You could see they polarize very nicely. There are cilia at the top, and they actually beat and they put out mucus, and they're very cool. And this is what we use for a lot of our experiments. And if you look at responses to respiratory viruses, looking at healthy kids versus asthmatic kids with two different strains of RSV, the asthmatic kids induce TSLP, the healthy kids do not. So that gives you an example right there of a difference between healthy bronchial epithelial cells and asthmatics. They have more release of cytokines that drive responses that may be involved in the response to RSV. Likewise, it's very similar when we look at rhinovirus. So we're now using this model system to try to ask what's driving the TSLP response, what's different between a healthy bronchial epithelial cell and an asthmatic. And it's the same result for IL-33 with rhinovirus. We're building mice now, we actually have them, where we can use the more relevant strains of rhinovirus or rhinovirus A. Its receptor is ICAM1, and the human virus does not bind to the mouse ICAM1. So we basically built a mouse where we can express human ICAM1 very specifically in airway epithelial cells or any other cell we want. And so we can now use the real rhinovirus, or the rhinovirus that's a problem, in mouse models to try to understand it. We're also building a mouse that will allow us to use a rhinovirus C. Yes?
Already sensitized, does it upregulate TSLP receptors?
In
other words, if these are allergic.
Oh yeah. So yes.
Yeah.
So and it does. This is an interesting, because it not only turns on the cytokine, but it also upregulates both components of the receptor. So you could get TSLP responses within the epithelium, and we're actually looking at that right now. So this is the closing thing. I'll be quick because it's almost on the hour. Looking at normal skin versus atopic dermatitis skin, the sort of brownish staining is TSLP in the epidermis of the lesional skin. So there's a lot of it. And so we wanted to ask, what does that do? So we built a mouse that allowed us to turn on TSLP specifically in keratinocytes. And this is a slightly different model. This uses doxycycline to turn on. So it's a tetracycline operator upstream of the TSLP gene, and then there's a protein that's made in bacteria called the tetracycline reverse transactivator. And basically it doesn't activate the operator until you give it tetracycline, or in this case, we use doxycycline. And then you get expression of whatever gene is downstream of the operator. You're taking advantage of what bacteria do. And so in this case, because we're using a modified keratin 5 promoter, it's only expressed in keratinocytes. And so we can turn on TSLP, to the amount we want by giving them more or less doxycycline. And we can take it, if we can turn it on, we can shut it off, we can turn it on. But what happens is these mice get atopic dermatitis. And so this is normal mouse skin on the left. This is the keratin 5 TSLP mice on the right. They get all the hallmark features of what humans get in at least as far as a mouse can do for atopic dermatitis. And so we wanted to use this model now in models of skin sensitization and then challenge in various other sites. So we set up this model where we either inject TSLP or use these mice to turn on TSLP and keratinocytes and give them an antigen. In this case, we do OVA. We've also done this with peanut, and it works the same. And then we either challenge them intergastrically if we're looking at a food allergy model, or intranasally for airway inflammatory model, and ask what happens. And if we challenge them in the airways, they get basically an asthmatic response. They've been sensitized in the skin, they now get challenged to the airways. And if we challenge them orally, they get food allergy and diarrhea. And they basically anaphylaxis, and some of them die. And so it's exactly what is seen in kids with food allergies, especially peanut is thought to be sensitized largely through the skin. And then we're from Gideon Lack in the UK. So now we have a model to look at what's involved in food allergy. And so we've done that. If we do it in TSLP, if we use self-antigen or don't give TSLP, we don't get a response. But you can see on the left panel, that's the percentage of mice that are responding with diarrhea. We get up to 100% after 4 or 5 challenges. And then the diarrhea scale is on the right, and that's basically 0 is normal, 1 is a little squishy, 2 is squishier, 3 is mostly liquid, and you don't want to know what 4 is. And so basically we get up to somewhere between 3, 3.5 for all of these animals. They get systemic responses, so there's circulating levels of IgE that's specific for ovalbumin, and we get systemic mast cell activation. So MMCP1 is a protein secreted by activated mast cells, and it's in the circulation. So most of the features that you see in a food allergic kid that sees their food, we can recapitulate in these mice. And they do, I'm not going to show the data, but they do anaphylaxis. They drop their temperature, they have vascular leakage, some of them die. And so we have a model both of food allergy and for anaphylaxis. It's TSLP receptor on dendritic cells that's important. So these are we have a flox allele for the TSLP receptor. We knock it out with CD11C, and we totally lose the response. And that's true for the local as well as the systemic responses. And what happens is these mice don't get primed. And so TSLP is basically driving the response in the skin. It's not, as I'll show you in a moment, it has nothing to do with the challenge. It's not involved in the challenge phase at all. It's like sort of like the GM-CSF in the cockroach model. TSLP sets it up in the skin, and then something else takes over in the gut. We don't know yet what that is, although we have an idea. We did it 2 ways. We sensitized and then treated with anti-TSLP, or we used a TSLP knockout, which we could use because we were actually giving them TSLP in the skin. And in both cases, when we challenged, we got full wild-type responses. So it's involved in sensitization. So we asked ourselves, well, what other cytokines are being made in the gut when they're being challenged? And this is just looking at a selection of genes. And there's some IL-13 and IL-4, which we weren't surprised about since it's a type 2 response, and those are effector cytokines. There's a little bit of TSLP. There's not much IL-33 transcription, but that doesn't really matter because it's released from necrotic cells. But we saw this little blip, and it looks little on this, but it's about 20-fold induction of IL-25 in the colonic epithelium. In these animals in the IL-10, we have no idea why there's so much IL-10. We haven't looked at that yet. So we said, well, maybe IL-25 is involved. And so we did that, a picture's worth 1,000 words. This is looking at a wild type mouse, and you can see there are no pellets in the colon, which means that basically it's washed free. So they have massive diarrhea. The IL-17RB knockout mice, which is IL-17RB is the IL-25 specific chain of the IL-25 receptor. So you knock that out, you don't respond to IL-25, you get a normal mouse. The knockouts sensitize just fine, but they don't respond to challenge. And that's looking at it more quantitatively. And then is IL-33 important? Because, you know, these three cytokines tend to work together. Right now, the model is TSLP is important in the skin, IL-25 is important in the gut, and they don't cross over at all. So is IL-33 involved? So we took ST2 knockout mice, so they can't respond to IL-33, and we did the whole model in those mice, you know, intradermal TSLP plus antigen, followed by oral challenge with antigen, and the ST2 knockout mice, no response at all. So basically, IL-33 is the alpha dog in this model, which has surprised the heck out of us. This is the same thing, except we wanted to look at priming. And what we see that in the skin, in the ST2 knockouts, even though we've given them TSLP, they don't prime, which suggests that IL-33 is downstream of TSLP in this model. Likewise, in the gut, we can give them, this is looking at the same, this is giving IL-33 in the skin in a TSLP receptor knockout. So in other words, do you need TSLP signaling and IL-33 signaling together to get a response? And the answer is no. So we can give IL-33 in a TSLP receptor knockout, and we get perfectly fine data. That's the model on top. That's looking at the colon, and then quantified on the right with the occurrence. There's basically no difference. So that suggests that 33 is what's actually driving this, and that TSLP is probably turning it on in some cell type in the skin. Likewise, in the gut, we allowed the mice to be sensitized, and then we gave treated with an antibody against ST2 that we know neutralizes IL-33 responses. It binds to the receptor, prevents IL-33 from binding. And in that case, we also got no response. And so it suggests that IL-33 is driving the response in the skin, but it's also driving the response in the gut. So it's basically in the skin, it's TSLP driving IL-33, and you get sensitization. In the gut, it's IL-25, because I showed you IL-25 is important. If you don't have a response, you don't get challenged. But it's turning on IL-33 somehow that we don't understand, and that's driving the sort of diarrhea and all the other downstream responses. And so, again, in this case, you can see this is a nice example of how these cytokines collaborate, and the collaboration is different depending on anatomically where you are. It's different in the skin versus the gut. And who releases IL-33 in the skin? We were betting it was going to be dendritic cells, but unfortunately it's not. This is using a mouse that's a flox allele of IL-33 with CD11 CRE. It's a wild type response, but then we knocked it out in keratinocytes and we lost the response entirely. So somehow IL-33, which is made by keratinocytes, but they also have the receptor, is feeding back and driving IL-33 expression in those keratinocytes, and that's driving the skin response. And we're still looking in the gut to see how IL-25 and IL-33 are collaborating. And then this is my final slide. Looking at, these are all the drugs, well, some of the drugs that are currently available to look at these responses. There's the anti-IL-5 receptor, which basically kills eosinophils. There's anti-IgE, which has been around for a long time, omalizumab. There's the anti-IL-4 receptor alpha chain, or dupilumab, that works really well, but it basically blocks the effector arm, as I mentioned, and you go off the drug and the response comes roaring back. There's anti-TSLP, which is in asthma, which looks really good. There's anti-IL-33, which is now in people, and mostly in asthma and in peanut allergy. Those trials are just getting started. There's an anti-OX40 ligand, because I didn't mention this, but how TSLP drives Th2 differentiation is it induces the expression of OX40 ligand on dendritic cells, and that is important for when those DCs then drive T cell differentiation. The OX40 ligand pathway drives it to a Th2 type response. There was an anti-OX40 that was used. It had a mild, if any, effect at all. I don't think it's being tested any longer. And there are other drugs that will be coming along for some of these other cytokines, and anti-kinase small molecules that are going against stats and against JAKs. And so this is the landscape. Hopefully that was clear, and I'm happy to answer any questions. Thanks.
While you have that slide up.
Yeah.
It seems to me, obviously the data don't support this, that if you block very proximately IL-33 or
Right.
TSLP, and you don't even know what TSLP does homeostatically, that's risky. If you use downstream like dupilumab, which works great, that might be a safer clinical strategy for human beings.
Probably. The alternative to that is that if you block very proximally, you have the chance potentially to change the response entirely, to basically tolerize, if you will. And there's a trial that was just done, we'll see if it works, by the ITN, the Immune Tolerance Network, in cat allergic people. Basically, because we know TSLP's up in those patients, and the idea is they were treated with sub-Q cat as a immunotherapy in the presence or absence of anti-TSLP.
This is Jonathan Corin.
Yeah, well, Jonathan was
yeah.
involved, yeah, it's called catnip,
Right.
if you want to look it up. And the idea is you give the anti-TSLP to dampen the inflammation that's seen, and that maybe would create an environment that's more acceptable, if you will, for driving tolerance, which is what you want to do with the immunotherapy. And the ITN's running a number of those kinds of trials in both allergy and in autoimmunity with these co-therapies with one drug that blunts the inflammatory response and a second drug that might be involved in driving tolerance. And catnip, they break the code in a month. So we'll find out if it worked. Matt.
And I mean, sort of building on that, I think that's a critical question of what's driving sensitization versus effector.
Exactly.
And I'm curious how much you can tell from your data, because also now there's a lot of interest in using this in very young children.
Right.
Some of these drugs in very young children to try to prevent
The dupilumab's down to 6 months old.
Right, is what they want.
Yeah. And, I think that's going to be what happens. I mean, if there's no side effects, people will get Dupi if they have ID, if they have, they're born with AD.
Right.
That's my guess. I wouldn't treat with anti-IL-33 then, because the brain's still developing. But Dupi is probably fine. We'll see about TSLP. It didn't work which was shocking, it didn't work in an atopic dermatitis trial, which is what you would think given what I showed you, but it was in adults and it's a different disease, so I don't know. And I was involved in some of the early discussions for the asthma trials with the anti-TSLP, and I didn't think it would work because my hypothesis, my bias, if you will, was that it's an initiating factor. So once you have disease and it's going, its job is done. But it turns out that the trial worked brilliantly, and so clearly we don't understand everything that TSLP is doing in that setting, and I suspect that's going to be true for IL-25 and IL-33 and GM-CSF and all these cytokines. We're only as good as the model systems that we use, and in the mouse models, it's all about initiation and sensitization with TSLP, and clearly that's not what it is in people. So,
I mean, yeah, I guess.
which is a good lesson.
Do you think it will ever be that clean? Like also your GM-CSF data
Yeah.
is very clean.
I know.
But
And these are SPF mice, so that's a real caveat. We really want to do these experiments in more, humanistic, if you will, mouse models, which is why we're setting up these wild mice cohabitation studies, because other people have used those on, pet store mice And and you get an immune system in those mice that resembles adult humans. In SPF mice, right off the shelf, they have an immune system that looks like a 2 or 3 year old infant, or toddler. I don't have children, so whatever you call someone who's 2 or 3 years old. So clearly you want to have model systems that more reflect the human situation, and we're hoping that will allow us to do that because things change. And it's driven partially by your microbiome, and it's driven by your environment, etc. So we're going to try to get closer to that.
The Th2A effector T cells.
Oh, yeah.
What are the prime drivers of that from the system?
We don't know for sure, but they're present in all of these animal models that I mentioned. So what Bill's mentioning is a type of Th2 cell, which Eric Wambre found at our place in humans, and Toshi Nakayama found in similar mouse models in Japan, which are the effector cell in allergy. So they're basically hardwired to make a lot of IL-5, IL-13, and IL-9 when they're triggered. They're found in people who are allergic to every allergen that Eric has looked at. The only difference in these cells in each allergen is their T cell receptor, but they're hardwired to respond exactly the same way, and they have incredibly high levels of ST2, and they respond to IL-33 very, very exquisitely. And they are the target. If you give immunotherapy to someone who's cat allergic or whatever, those are the cells that are targeted. They get killed. You go off the therapy, they come back. And so the nice thing about knowing what cell is driving the response is that they become both a therapeutic target, if you will, but they also become a biomarker for whether the therapy is working or not, and whether the allergy is coming back when you get them off therapy. So now Eric is working with a bunch of people to try to understand ways to get rid of them and have them not come back. But they are, what triggers their development, if you will, because they have aspects of a garden variety Th2 cell, but they're also very different. If you do single cell sequencing, you can clearly tell a Th2A from a garden variety Th2 cell. They do further differentiate, and what drives that is not at all clear. And that's another obviously important thing to try to understand, because maybe you can target that aspect and not even get to the Th2A differentiative step at all if you can intervene there. And that's being actively looked at right now.
So can we use them? Are people developing that as a biomarker after we have people on conventional
Yes.
immunotherapy?
Yes.
And we can say to them,
It's
the level is
working.
low enough,
Yeah.
we can stop your immunotherapy?
Yeah, but then they'll come back. But yeah, so Eric has worked it out now that with a 1 cc blood draw and 5 antibodies, you can look at Th2As. So it's down to being technically good for a clinical lab. Yeah. And you don't have to know what the allergen is. You just have to know that the cells are there or not. If they're there, you're allergic, basically. And if they're not there, you're not.
Well, thank you immensely.
Oh, sure. Thanks.
Brilliant talk.
So that was okay.
Bill, I wanted to ask you, I was,
okay?
I'm inherited status.
Yeah.
And it was supposed to happen.
Thank you.
on August.
Brilliant. Now we'll go back to sleep.